Friends, welcome to Biology Made Easy. Today in this video we'll discuss about incomplete dominance. So what is this incomplete dominance we will discuss, but before that let me tell you one thing. When Mendel conducted his monohybrid crosses in the F2 generation, he obtained the phenotypic ratio as 3:1 and the genotypic ratio 1:2:1. But later on, when post-Mendelian scientists started working on different monohybrid crosses in different plants, they found that the phenotypic ratio is modified. It is because of the different interallelic interactions, and sometimes it is also intergenic interactions. So the post-Mendelian disorders we can classify into the post-Mendel, or you can tell gene interactions. These are mainly of two types. It may be intragenic, or we may call it as interallelic. Because within one gene there are two alleles, so between the alleles, or it may be intergenic, that is between the genes, or you can call it as non-allelic. These type of interactions are found. What are the examples of this interallelic or intragenic interactions? It is incomplete dominance, then the second one is codominance, and the third one is multiple allelism. These are between the different alleles of the same gene, but non-allelic or between the different genes. These are again of different types. One, it may be due to complementary genes. Number two, supplementary genes. Number three, it may be due to epistasis, or it may be due to duplicate genes, or it may be collaborator genes, or it may be polymeric genes. These are the different interactions between gene and gene, that is non-allelic. But these are the different interactions between different alleles of the same gene. So here incomplete dominance, codominance, and multiple alleles is coming. And from this, today in this video, we will discuss the incomplete dominance. So when the question of incomplete dominance is coming, what is the meaning of it? As all of us are aware that when Mendel conducted the monohybrid crosses, having one dominant and one recessive character, in the F1 hybrid only a single character appeared, and the character which appeared is known as dominant character. And this dominant character was expressed whether it is in homozygous condition or in heterozygous condition. For example, in one of the experiment between tall and dwarf plant, in F2 generation he obtained this tall plant, and also this is also tall plant. That means when they are present in homozygous condition or whether they are present in heterozygous condition, in both conditions the capital T is complete dominant over small t. That is only one character. This is known as complete dominance. But we are discussing incomplete dominance. Post-Mendelian scientists, when started working on the other organisms, they found that one of the allele is not completely dominating the other one. There is neither complete dominance nor there is complete recessiveness. Both the characters, they blend together. The characters blend together, or we can call that both of them are mixed together. So this blending of the two characters, or the mixing of the two characters, is known as incomplete dominance. This is known as incomplete dominance, or we can call it as also the mosaic or blended or partial, or you can call it also intermediate also we can call it intermediate. That means say in this case one allele is not completely dominating over other, and as a result what happens in the F2 ratio, the monohybrid cross, the F2 ratio is 3:1, the phenotypic ratio. This phenotypic ratio is modified to 1:2:1. That means it is equal to that of the genotypic. The genotypic ratio is usually 1:1, while the phenotypic ratio is 3:1 according to Mendel. But here the genotypic, phenotypic, both the ratios are coming in 1:2:1. So let us give some examples. So here we will give 3 examples. One example is that of Mirabilis jalapa or 4 o'clock plant. Other is Antirrhinum majus, or we can call it as dog flower or snapdragon. And another example we can take from Andalusian fowls. So the different examples are, suppose example number 1. The example number 1 is Mirabilis jalapa. This is also known as 4 o'clock plant. Now in this case, when a cross was made between pure red, which is the genotype is RR, with pure white, which is RR. So naturally, the F1 hybrid will be capital R and small r. If it would have been Mendel's cross, then this F1 hybrid would have pure red, but not here. Here, all F1 hybrids were pink. That means the red was not completely dominant over white, nor the pure white is completely recessive to the red. That means here both the characters blended together, they mixed together. Intermediate character came, that is pink. Now when two such F1 hybrids were interbred, that means RR and RR, that is when two are intercrossed, what happened in F2 generation? This is RR and this side it will be RR. So in F2 generation, capital R, capital R, capital R, small r, capital R, small r, small r, small r. Now the capital R, this will be pure red. Pure red. The capital R, small r, this will be pink. And small r, this will be white. That means pure red is 1, then pink 2, and white 1. That means instead of 3:1 ratio, now the phenotypic ratio is 1:2:1. If we consider the genotypic ratio, then RR, they are also in the ratio of 1:2:1, because this capital R, small r 2. That means both phenotypic ratio and genotypic ratio are in the same ratio of 1:2:1. This is the case with that of the Mirabilis jalapa. Now in the same case, if you take this second example, in case of Antirrhinum majus, which is also known as dog flower or snapdragon, the same case. The same case means when you will take cross between the red and white, the F1 hybrid will be pink, and again the F2 generation this will come. The same result No problem. Now let us take the example from that of Andalusian fowls. Example number 3. Andalusian fowls. So in case of Andalusian fowl, a cross was made between pure black. Pure black. It is designated, the genotype is capital B, with pure white, and that is small b. So the F1 is capital B, small b. If it would have been Mendel's monohybrid cross, then it would have been black, but not here. Here all the F1 hybrids were blue. Well, blue. And when two such blue F1 hybrids were intercrossed, then this is capital B, small b, this is also capital B, small b. So from this Punnett square, we'll get capital B, capital B, capital B, small b, capital B, small b, small b, small b. Now you see, this is pure black, that means black. And when these two, that is blue and white, they are coming in the ratio of 1:2:1. Because this is black, these two are blue because they behave like the F1 hybrid, while this is white. Now we will come to the genotype. In a genotype, this is BB, capital B, small b, and small b, small b. So these are also in the ratio of 1:2:1. That means both phenotypic and genotypic ratio are same, 1:2:1. This is very important question also for examination point of view. So these are the different examples. Now we have to explain why such things take place. Let us explain why such things are taking place. Now, as all of you know, that each gene, it contains information for the expression of a particular character. We have to give the explanation for it. That means each gene, it contains information for the expression of a particular trait. Now, this each gene, when it is present in 2n condition, it has two alleles. It has two alleles. Each gene has two alleles. Now, what happens? The normal allele, the allele which is normal, usually the normal allele transforms the substrate S into products, but the allele, if modified, if one of the allele will be modified, what will happen? It may give rise to normal or less efficient enzyme, or it may give rise to non-functional enzyme, or it may give rise to no enzyme. These are the probabilities. So, if the first case takes place, that means in the first case, it is equivalent to that of the unmodified allele. But what about the second and third case? In the second and third case, what will be the phenotype? That depends upon how these are functioning. The phenotypic effect the phenotypic effect depends on functioning. That means what type of product they are producing, that depends upon what type of phenotype will be expressed. So in the first case it is equivalent to unmodified allele, while these are different. That means the first allele, or you can tell the unmodified allele, the unmodified allele is dominant, and the modified allele is recessive. That is why the behavior becomes different. So these are the three examples. Let us give one fourth example and more interesting. Sometimes a gene produces a product, and this product has the product has a different effects in the same plant. Let us take the example of starch grain synthesis in case of a pea plant. The size of starch grain in case of pea plant. Example number 4. Size of starch grain in pea plant. So here I already have told you, a single gene produces a product which has different effects. So in this case there is one gene that is responsible for the synthesis of starch. Now when this gene is present in a double homozygous dominant form, then it gives rise to large size starch grain, and when mature they are round when mature. Now the BB, it gives rise to small size starch grains, and they are wrinkled when mature. But capital B, small b, it gives rise to intermediate starch grains, but these are round when mature. Now you see, here there are two characters. If we take the round and a wrinkled character, they will come in the ratio of 3:1. But if we take the size of starch grain, then it is coming in the ratio of 1:2:1. Both the phenotype as well as these are the three genotypes, and these are the phenotypes. They will come in the ratio of 1:1. That means it is the same gene, but when it has effect on the shape of the seed, it is dominant and recessive. That means here this gene is the capital B is completely dominant over small b. But when the same gene has effect on the starch size of the starch grain, then it is not completely dominant, it is incomplete dominant, so that it is giving the ratio of 1:2:1. That is the same gene has two different effects. So this is about incomplete dominance. Now let us discuss MCQ. So MCQ number 1, 1:2:1 phenotypic and genotypic ratio is found in first choice complementary genes, Second choice, blended inheritance. Third choice, multiple allelism. And fourth choice, pseudo alleles. Then the second question, incomplete dominance is an exception to first choice, law of dominance. Second choice, law of segregation. Third choice, law of independent assortment. And fourth choice, all of these. So these are the type of questions which are usually asked in all type of national examinations. I hope now you have a very clear understanding about incomplete dominance. Thank you very much for watching the video. God bless you. Have a good day.